and it's on the role of plant breeding in native seed stock, defining the challenges and opportunities. So I'm Lee Moulton. I'm one of the steering committee members of the GRF, and a little bit of a background on what the GRF is. Our mission is to provide a forum for information sharing, tool development, research, and education to promote and support conservation and effective restoration of native grassland ecosystems in Alberta. And this work was begun in 2006 as a collaboration between various provincial agencies, ranchers, conservation organizations, First Nations, industry, ecologists, and reclamation practitioners. So in an effort to support this work, the GRF offers these free webinars. We have one more for the season on April 15th with John Acorn, also known as the NatureNet from the University of Alberta. And he'll be speaking on the conservation value of erosional landscapes. We also offer a few different courses every year, one of which is our fall range health assessment training course. And this is a 2-day course, usually held in September. And it has a classroom portion and a field portion, which we go through the range plant community guides and recovery strategies, as well as the field-based assessments. And that is usually held at near Brooks at Antelope Creek Ranch. We also have kind of a 1-day classroom version of this and before the field season, and it's actually being held on the 26th of March. So next week you can still register if you are interested in that. I believe it's still open. And that's in Cochrane at the Glenbow Ranch Schoolhouse. This is really useful training. This one-day portion for kind of, if you're going to be doing any work in grasslands during the year, it's a nice kind of brush up on your skills before field season. As well as we usually have, in November, we have our fall information session as well. So that's in November. So watch for some of the details of that. If you haven't already, join our mailing list. You can go to our website and find all the contact information there and all the other events that we host. All that is available on the website. So for today, I'd like to introduce Dr. Sean Asselin, who is a perennial crop breeder with Agriculture and Agri-Food Canada, specializing in the breeding and genomics of native rangeland plants for their maintenance and integration in agro-ecosystems in support of ecosystem services. His lab in Swift Current focuses on population level variation in stress tolerance, identification of genes associated with adaptation to climate, production challenges, and genotype, environment interactions of native plants to support breeding of native species for forage production and reclamation end uses. So he's gonna be talking to us about the native forage integration research at the Swift Current Research Center. As well, for the duration of the presentation, please leave your cameras and your mics off. And any questions that you have during the course of the presentation, please put them in the chat and we will go through them once the presentation is done. And with that, Dr. Asselin.
All right, thanks, Lee. So I'll get my screen shared here. We can get started. Let's see. And get PowerPoint up there.
Got your presenter screen again.
Oh yeah, I'm just gonna swap it over. Okay. There we go.
Perfect. So yeah, you're all set.
All right. Perfect. Thanks everyone for joining today. So my name is Sean Asselin. I'm a research scientist with Agriculture and Agri-Food Canada, based out of Swift Current, Saskatchewan. And today I'm going to be talking about a little bit about my research, which is focused on the use of native species in agriculture, specifically from a plant breeding perspective. So just to get started, a little bit about where I work. I work for AFC out of Swift Current. We have a research and development center here just on the edge of town. And this is a unique facility within Canada because it houses Canada's salinity screening facility. So this is a hydroponics facility that has traditionally been used to develop salt-tolerant forage crops. but we also use it for a variety of abiotic stress testing projects like drought tolerance. And in terms of the land base, we have a home base of 930 hectares here in Swift. 435 of those hectares are in tame pasture, but we also have a considerable 140 hectares of native rangeland on site that are used for various beef and forage grazing projects. We also have up-to-date greenhouse, grow chamber, and animal care facilities. And we are set up to do, well, soon to be set up to do NAR. We are set up to do hyperspectral imaging and drone imaging of field plots, as well as analytical chemistry on site. And we have a number of researchers, myself and my colleagues here, that have been working on projects that relate to the integration of native species into agricultural production systems. Give you an overview of kind of some of the local work that's been happening within AFC, specifically in Saskatchewan. Here in Swift Current, we have my program, which is the Native Forage Breeding and Genomics program. We have my colleagues, Klilu Alimu, who does ruminant nutrition. So he's been doing work on species such as prairie clovers, looking at the impact of condensed tannins on ruminant diets, potential implications for reducing methane emissions, and other nutritional benefits to animals. We have Alan Awasa, who's our grazing management scientist here, and he's been doing work on native and tame forage mixtures, as well as rotationally grazed native stands, and looking at the productivity and environmental benefits of having native species in rotation. And he's had projects that go back actually to the 90s here. So we have a number of long-term experiments that have been run on native species here. We have Francois Test, who's a relatively new scientist here, and he's looking at plant-soil feedback, plant-soil interactions. And we're working together on a few native forage establishment projects. And then after the res staff, we have our biologists. So we have Dustin Ostrander, who's our rangeland biologist here, former PFRA, as well as Shanna Quilicini. And she's the lady standing on the yellow box in the photo there. And she runs the salinity facility here, but she has a background in forage production, and her MSc was specialized on the use of native species or in mixture with tame species. Up in Saskatoon, we do have some support through the PGRC. So this is Plant Genetic Resources Canada, and this is the gene bank for all, genetic materials in Canada related to plants. That's led by Axel Diedrichsen. He's the curator. We also have support from Tina Bunderock, who is his biologist, as well as Cam Cater, who is another rangeland biologist in Saskatoon, who's going to be helping us put in various plots. Up in Beaver Lodge, Alberta, we do have Nitya Canal, who is a forage seed production and agronomist specialist. And he previously worked in the program here in Swift Current under my predecessor, Mike Schellenberg, and has experience with native plants. And so that's kind of the local group. I will mention, though, that we are highly networked, both within AFC and other Government of Canada departments, as well as externals. We do quite a bit of work with the universities of Manitoba and Saskatchewan. And we have larger projects that are shared through departments, such as the Genomics Research and Development Initiative, and large collaboratives with the universities through Genome Canada. Okay, so now to get into some of the context of native seed and how it's used in forage production systems. So native species in the West are primarily valued for their late season forage. On the side of the screen there, I'll just point out that graphic is the forage productivity of various crops in Saskatchewan throughout the growing season. And you can see that in the later growing season, we do have a lull, kind of a drop off in productivity of most of our introduced tame species. And that actually happens to coincide when a lot of our native cool season grasses tend to be at peak productivity. A lot of our native species tend to reach maturity later in the growing season and tend to have better forage quality going into August and September. So there is that niche there for forage productivity in rangeland systems and tame systems. Breeding work on native species for forage production has been going on since about the 1920s here. Early work was focused on some of our self-pollinated grasses like slender wheatgrass and green needlegrass, which are both native to Western Canada. And work has continued to kind of off and off in spurts from the 1920s until now. Now, the reason why native seed is so important in these production systems is most of our remnant prairie is preserved within privately owned rangelands, and they are a major carbon stock or carbon major carbon holding capacity, I should say. And about 34% of terrestrial carbon stocks are found in prairie ecosystems. And avoiding the conversion of both native and managed grasslands really represents a large opportunity for agriculture in Canada to mitigate its CO2 emissions. So those are some of the reasons why we're interested in using native species. But of course, there's also biodiversity benefits and a number of ecosystem services that are unique to various native species that do help with productivity that kind of drive that interest in native species. Okay, moving into plant breeding. See here. So really, a major part of my work is focused on the adaptation of native species. We have a lot of species that have been expatriated across the prairies, so reintroducing them can be a challenge if there are substantial interactions with environment. These are known as G-by interactions. And just as an example, the background on the slide here, this is a Martin nodding brome nursery that went into swift current in 2020. And we had a major winter kill. We lost about 95% of the nursery. You can see there's only about 7 plants on the screen there, and that's all that was left because those were the plants that happened to have enough cold tolerance to persist. The rest of the population of about 200 plants did not persist, and that's due to a genotype times environment interaction, and that's really what we're trying to avoid when we develop new seed sources, is we want materials that are going to have the adaptive capacity to grow in their targeted environments to reduce genetic offsets. And this is classically known as G-by E interactions. So plants can react kind of one of 2 ways in different environments. They can have what's called phenotypic plasticity. So that means, all genotypes of the species, they're broadly adapted enough that they should be able to grow in many different environments. There's no real interaction with the environment. In the middle graph here, you see that's a genotype times environment interaction where one genotype has a decline in productivity or decline in survival, decline in the trait that we're interested in one environment relative to the other. And then other genotypes might have better productivity, might have better local adaptation to that environment. And then sometimes what we see is, one genotype might have better performance in one area versus another, and then the other genotype might just be stable across all environments. Okay, so now going into kind of the role of plant breeding for native species. Really, the main characteristics we're interested in are establishment, persistence, seed production, and then forage yield and quality. There's a number of benefits of selecting for these characteristics in native forage species, and it really relates to kind of their agronomic management. And I'll show a few examples in the next slide of why that really hits home. We also work on challenging traits of wild species, primarily related to seed quality. We do selections for improved viability or reduced seed dormancy in select species. There has been some work done on some native species relating to shattering and asynchronous maturity because these are characteristics that made seed harvest a lot more challenging. We're not looking at those necessarily in my program right now because there are ecological functions that are associated with shattering and timing of maturity. And we don't necessarily want to breed those out of materials that are going to be going in the field because it might have negative impacts over time. But we're really focusing on the establishment, persistence, seed production, yield, and quality aspects. And really, this is to reduce the high production costs that's associated with native species. Just to give you an example of 2 native species that are commonly used in mixtures, June grass and slender wheatgrass. They vary quite a bit in their biology. June grass is commercially, it's less common. It's a relatively slow species to establish, and not all ecotypes actually produce seed every year. Sometimes they are alternate bearing. It's also a cross-pollinated species, so you can deal with issues with not enough individuals being present in the population, and you result in poor seed set because there's just not enough diversity. Something like slender wheatgrass, it's been a species that we've been working on for about 100 years. It is a easy to establish species, very easy to harvest mechanically, and it's also a self-pollinated species. So if grown in isolation, it will set seed. And just looking at the price per pound of seed, June grass is at $67 a pound. Part of the reason for that is sometimes we have to go to wild harvest for that species because it is hard to produce commercially. And then we have slender wheatgrass, which is very easy to produce at a commercial scale. So we do see a corresponding lower seed cost there. And really the point what I'm trying to make here is that breeding and agronomic research and development can reduce the cost of production for these species, and that ultimately trickles down to the end user. And for comparison, alfalfa, high quality elite alfalfa lines are about, $2 to $3 a pound, and really those are the species that we are competing for acres with. So reducing the cost of seed production is a major target of the program to make native species more competitive with some of our introduced and tame forage species. Moving into how we actually do this, so we go through repeated cycles of selection to develop populations. This all starts with a base population, so this could be wild harvested populations. Typically, we try to use multiple seed sources when we're making our selections because we don't want to constrain genetic diversity. And what we do is we collect our materials and we divide them into genetic units. In this example here, we're talking about half sibling families, so this would be seed off of a maternal plant. And then we do evaluations, typically in a single site over multiple years. At later stages in the breeding process, we do multi-site testing, but initially we usually test materials here in Swift Current for 3 to 4 years, make selections of our best materials, allow those to open pollinate to create a new population, and then we repeat this cycle until we get material that is suitable for our end use. So if it's reduced dormancy, better winter survival, better establishment characteristics, those types of things. And one of the things we have to be really careful in this process is making sure that we maintain genetic diversity. We don't want to select too hard for any one particular trait within a given population because it can exhaust the genetic diversity. And then when we lose our genetic diversity, we often lose our adaptation capacity to new and different environmental stresses. But there are some ways of managing this type of making selections while maintaining genetic diversity. And the way that we do this is using a technique called among and within family selection. And I'll just go through kind of an example of how that works. So when we evaluate using among and within family selection, we take multiple families of the given plant, and these can be populations that come from different parts of Saskatchewan, Manitoba, and Alberta. And we evaluate first the family level, and we either do that using space planted nurseries. So these are plants that are put on 1 meter rows, spaced apart from each other, and we just make selections for the average of those plants from a given half sib family or full sib family. We can also do this under rows where the plants are seated right next to each other, and that gives them a little bit more competition, and that's more realistic. Or we can grow them in swards. That would be like you would see in a standard forage situation. And we start off by collecting data on all the plants and genetic units, and then we get averages for each one of the families. From there, we make selections from the families. So in this one, we're saying that family A, B, and D, those are the best families to keep. And this could be for any number of traits. Just an example, winter survival is a good one. If we have 5 seed sources and in the first winter we lose, 1 or 2 of them, we don't continue on with that family. And then we move on to the plants within the family to make a further round of selection. And this is what we end up with is we select the individual plants. Those individual plants we'll either dig up or clone and put them to what's called a polycross. So this is how we make crosses to create new combinations between the parental materials. Basically, a space planted nursery of the best genotypes that have been replicated, and we allow them to open pollinate. And then we can go on to field testing and large-scale GBIE testing from this point, or we can do repeated cycles of selection. So on the right-hand side of the screen, that's just a photo of one of our polycrosses of northern wheatgrass growing here in Swift. And on the next slide here, this is what that polycross looks like, what's in a sward type growing condition. And typically, we would have this replicated at multiple sites across Canada to look at how well the materials are performing. And typically this is the step that we take before we go to registration. Now, I just want to point out that when we're doing this recurrent selection, our accuracy is limited by the genotype times environment interactions and trait expression. So even when we make selection for a given trait, we don't get it we don't always get a dramatic shift to the end product that we want ultimately. Often it takes multiple cycles of selection based on the trait and how much standing genetic diversity there is within the initial population. And as we make selections, typically our gains under selection tend to decrease over time because genetic diversity for that given trait gets exhausted over time. So a population that, if we're making selections for reduced dormancy, it might start at, 75% dormancy, going down to 60% after, first round of selection. But then, when we get to those further round of selections, we do have diminished returns because genetic diversity does starts to decline for that given trait. Now, I'll point out that in theory, the trait under selection is independent of adaptive traits. That's one of the things we try to avoid. And so when we see this decrease in variation, it's based on a single trait, not on all traits. So just to make sure that's clear. But we go through repeated cycles of selection, and there's always overlap between generations for what we see in terms of phenotypic expression. Now, moving on to which species we're focused on. In the core breeding work in my program, about 80% of the work is focused on native species. So in terms of our grasses, we've got a number of cool season grasses like northern wheatgrass, plains rough fescue, and green needlegrass that we're doing work on. In terms of warm season grasses, we're focused on prairie sandreed and side-oats grama for breeding. We do a little bit of GBIE testing in other species. For legumes, we've been doing a lot of work on purple and white prairie clovers. We've actually been doing quite a bit of genomics work in these species as well, which I'll talk about a little bit later. We're also looking at big leaf lupine currently, as well as Canada milkvetch. And we're also developing seed sources for various shrubs and forbs, including winterfat. And we're just starting to do a little bit of preliminary work on Maximilian sunflower. And the other 20% of the program is focused on tame forages. So we're doing work on intermediate wheatgrass, also known as kernza. And we've been doing selection for salinity tolerance in that species. And we're also looking at that kind of mixed use potential of that species because it's been considered a candidate for the development of a perennial wheat type product. So we're interested to see it in that dual use potential. But ultimately, the core breeding work in the program is focused on adaptation, establishment, and persistence under semi-arid conditions, where some of our tame species tend to struggle with recurrent drought and cold winters. So that covers kind of the core concepts of my program. And now I'm going to just kind of give you an overview of some of our current projects that we're focused on here. So the first is the beef cluster. And this is the core breeding work, and this is a collaboration with an AFC and with the University of Manitoba and University of Saskatchewan to develop germplasm for forage production. And really, we do evaluation of new populations. We screen for new traits as part of this work, and we make selections and produce crosses like the northern wheatgrass polycrosses I talked about earlier. A big part of this work is pre-registration variety testing. So within the forage world, we do develop varieties that are protected under plant breeders' rights because they contain either specific traits, or we want to be able to make sure that we can confirm the origin of the materials or the zone of adaptation. And we do pre-registration variety testing to look at establishment, adaptation, persistence, forage quality, biomass, and yield, as well as the stability of these characteristics across multiple sites. Currently, we're doing multi-site testing for switchgrass. And on the bottom right there, you can see that's one of our switchgrass plots here in Swift Current. And you can see we have some lines that are doing okay, some that are really struggling. Swift current happens to be outside of the normal range of adaptation for switchgrass. So this is somewhat to be expected, but we do see major genotypic differences between our seed sources. We're also doing GBIE testing on northern wheatgrass, which I showed in the previous slides. And we're looking to start prairie sandreed, side-oats grama, and little blue stem this year, as well as plains rough fescue. And this is for selection track materials. Moving on to a little bit more on breeding in terms of the sites that we use to test. So because we collaborate strongly with the forage breeding programs, currently we're testing materials with Dr. Bill Billigatu and Doug Catani at the University of Saskatchewan, Manitoba, respectively. Annie Claessens out of Quebec has been testing some of her materials. She's also the breeder that has been developing the switchgrass lines that we're testing. And we also have Hari Pudel out in Lethbridge who does testing for our materials as well. And we have Yusuf Papadopoulos in Kentville. Currently, we're not testing each other's materials just because the zone of adaptation is just for a lot of these species is constrained to the prairies. But we do work together on a variety of initiatives as part of the beef cluster. Okay, moving on to the second project. So this is GG4GHG. It's grassland genomics for greenhouse gas mitigation. And this is a major Genome Canada project that's being co-led between the University of Saskatchewan and AFC here in Swift Current. And this project has involved the intensive sampling of intact prairie, as well as restored and rangelands from across Western Canada. John Bennett's group at the University of Saskatchewan, he's the co-lead on the project, and his group has done a tremendous amount of sampling both intact and restored rangelands, looking at soil quality parameters and soil microbial health. Some of the targets that we're looking at in this project are evaluating ecosystem services associated with prairie plants. So seeing for a producer, what is the benefit of including these species within a forage mixture? And we're looking at both the economic aspects of it, so price of seed, ease of establishment, but also looking at the ecosystem services associated with those species. So what are these individual species doing to the soil? Are they fixing more carbon or nitrogen? Are they providing habitat for beneficial insects and other kind of downstream ecosystem services? My part of the project is really looking at plant genetic diversity and how that relates to abiotic stress. So as part of this project, we've been looking at calculating what are called genomic estimated adaptive values. So looking at genetic variation in different prairie species and how those and looking at the genetics of different populations and how those populations associate with environment to develop basically scores to see how well different populations fit in different environments based on their genetics, or for future materials if they show a good fit or not. To a given environment. So this is very similar to the concept of genomic selection that we use in most crop species, but it's focused on environmental adaptation as opposed to things like wheat yield or disease resistance. Some of the work we're currently doing includes environmental genome-wide association studies. So this is looking at genes associated between climate and genetic variation. Again, potential to be used as predictive markers for adaptation to certain environmental stresses. We're also looking at evaluating the population structure of a number of species to see if we have distinct populations across the prairies or across North America. And this is going to help us direct the way that we develop populations. So do we need to have 25 different seed sources for a given growing region, or is it one or two, or do we have species that are broadly adapted where this this development of multiple populations is needed or not? So those are some of the kind of questions we're getting at. We're also been doing quite a bit of work on developing reference genomes for native species. In terms of the target species that we're looking at right now, as part of GG4GHG, we base the species that we selected for this project on functional group diversity. So we have a cool season grass, which is green needlegrass. That's on the top left of the four images there. Prairie sandreed, which is on the top right. Maximilian sunflower, which is the big banner shot at the bottom. That's a photo from my PhD. I did sunflower or native sunflower breeding for my PhD project. So Maximilian sunflower is near and dear to my heart. We're doing work on Lewis blue flax, which is on the bottom right-hand side there, as well as purple prairie clover, which has been under development here for a number of years in Swift Current. We're also doing a little bit of work on white prairie clover because we were able to bring in some sequencing data from some previous projects. So we're doing a little bit of work on the environmental GWAS of both purple and white prairie clover. And there's some interesting comparisons that we started to do there, which I'll show in a few slides here. Going into some of the work that we're actually doing in this project in Swift Current. So I mentioned the salinity facility earlier. One of the great things about this facility is that it's a purely hydroponic facility. So we grow all the plants out in a inert silica sand that has a coarse grit to it. And so it drains quite well. So we're able to control the water levels in this facility really, really effectively. So looking at plant response to drought is one of the things that we've been doing in this project in the facility. These are the 5 target species from the Genome Canada project growing here. And on the top left here, that's just kind of early establishment characteristics. Bottom left, those are the plants at kind of peak biomass before we turn the water off. And what we did in this project is we evaluated about 1,700 plants under drought stress. So we did a progressive drought stress trial where we reduced water by 25% each week over the course of several weeks, and then entered into terminal drought. And we took a look at physiological response to the plants over this period. And at the end of the test, we harvested the biomass and then did regrowth ratings to look at what materials were able to survive the drought and come back. And you can see green needlegrass on the right-hand side of the screen. That's some of the regrowth. And the reason that we're collecting this data is we're looking at the drought tolerance characteristics of these populations to develop genetic markers that can help predict response to drought. And in the next slide here, this is kind of what that looks like from a genetic mapping kind of point of view. This is a GWAS that we've done, so a genome-wide association study, where we looked at the association between genetic variants, so genetic markers that are called SNPs. And in this slide, we actually looked at environmental variables. So looking at how closely associated were genetic variants between populations associated with different climatic variables. And one of the things that we found is that in prairie clovers, both purple and white, we have a major region associated with cold temperatures. And these are actually associated with quite a few different bioclimatic variables. So for context, both of these species, we collected populations from across North America, either from sampling in Western Canada or from using the USDA gene bank to receive materials. All these materials were geo-referenced, so we know where they come from. Then we genotyped them, and then we looked at the climates of where all these plants came from to make these associations. And this is basically a way of helping us predict how these plants will respond to cold tolerance. And we still have to do confirmation testing of if these genes can be used as selectable markers for cold tolerance, but this is kind of early stages. And we're doing this for a number of traits, so a number of bioclimatic variables, as well as traits like drought tolerance characteristics that we're screening in our greenhouses. And we actually found that in both the purple and white prairie clover, it does appear that the common gene or common region that's under selection for these traits, which is really cool. So we're hoping to get a paper out on this in probably the next probably 6 months or so. Moving on to the next project, I am working with Francois Test here in Swift Current on winter fat, purple prairie clover, and Indian ricegrass. And we're looking at various seed treatments that can be used to help improve their establishment. One of the challenges with winter fat, which is it's a native shrub, it has a very fluffy pappus, so it's got a fuzzy seed. These seeds tend to get stuck in machinery when you try to put them through the drill. So we've been looking at various seed treatments that could improve the flowability of the seed, so it's easier to establish them. We've got the 3 species, but winter fat is really kind of the big focus of this project because it is a really high quality forage in the winter months. It has about 14% protein content. So there's a lot of really good potential for it to be used as a winter grazing species. But the challenges with it are establishment and low viability of the seed and storage. And those are some of the things that we're starting to look at in this type of project. Currently, we've been looking at pelleting the seed using different seed treatments, so it flows through the drill easier. We've been looking at flaming, gibberellic acid coatings to help with emergence, as well as microbial inoculants that may help with the establishment. The next species, and this is a bit of an, well, I wouldn't say it's quite a new project because it's kind of a continuation of a previous project. Those of you who may have seen my presentation last year, I talked about vigor in prairie legumes and some of the work we were doing on purple and white prairie clover to make selections for vigor and establishment using camera systems. And that slide at the top there, that's actually a composite image we developed using Raspberry Pi cameras in the greenhouse. And we developed an automated system to basically do time-lapse photography on our emerging seedlings and calculate the percentage of green in each pot. And this helps us get a better estimate of vigor in a really high throughput way. We're continuing some of that work, and now we're extending it to seed traits. So we have a new project that has just started that's working on purple prairie clovers, American vetch, as well as big leaf lupine. And we're screening seedlings using a hyperspectral camera and to develop a non-destructive imaging pipeline. And this is a technique that's been applied in some of the tame forages. So I have an image there from a paper where they did this in alfalfa. And basically what they did is they're able to scan the seeds and look at their spectral profile and then correlate that with seedling performance. So is the seed alive or dead? Is it a hard seed? Or is it something that's very high or very low vigor? Characteristics of the seeds, such as chlorophyll fluorescence, are major targets that we're looking at in this project because it often correlates with seed health. So we're just starting out that project this year. Another project we're looking at right now is the evaluation of forage characteristics of lupines, so specifically Lupinus polyphyllus. So this is a native species, but it's really limited by its alkaloid content. So it contains bitter anti-nutritional compounds, so it's not recommended to use this as a forage crop. But there has been some evidence that it has decent salt tolerance, drought, and acid tolerance. The materials have been performed really well here in Swift Current, so we're interested in doing further work looking at the evaluation of biomass and forage quality. We're also currently doing work on seed yield and yield components. The seed of polyphyllus has a really high protein content and high energy content. Also a really good high quality amino acid composition, so it is comparable to soybean. And it probably has a wider adaptation than soy to Western Canada. So we're interested in developing this species as kind of a forage crop, either used as outright as a forage of the biomass or for seed production and using that seed as a protein supplement. But it's just another option for the use of native species. But really, we have to get at the alkaloid content in this species to decrease that. We know we have genetic variation in this trait, but we need to develop some screening protocols for that. And that's going to be part of the last project I just spoke about on seed phenomics. Okay, I'm going to talk about one more project here, and then we can go into the question period. So this is the newest project my program's starting this year, and we're looking at the ecological combining ability of native legumes with winter cereals. So in the United States and in Europe, we have a number of forage crops that have been successfully used as living ground cover in winter cereals. So that's something like red clover, alfalfa have been used quite successfully in combination with species like winter wheat. And this provides a number of benefits in an agroecosystem. The trait that we're really interested in is nitrogen fixation for the winter cereal crop, but as well as weed suppression. So this helps decrease the amount of herbicides, pesticides that need to be used on a crop. So it has that environmental benefit. But there's also synergies between some of these winter cereals and our native species. As I mentioned in one of the first slides, a lot of our tame species, they tend to decline in quality in late summer, but that's when a lot of our native species, including the native legumes, do have higher productivity. Species like the prairie clovers, I mean, their biomass production usually tends to peak in around August to even September. So we're interested in really intercropping these with winter cereals to kind of not only for the environmental benefits, but also to maximize that kind of potential forage production. And in terms of species, we're looking at a pretty wide list. Basically, every commercial native legume we can get our hands on that's adapted to Western Canada. So we've got prairie clover, sweet vetches, lupines, various astragalus, so milkvetch, as well as local weed species. Now, not all these species obviously are going to be used for forage. Something like local weed, you don't want to be feeding to your cattle, but it has that potential to be used as a ground cover for those environmental benefits in situations where a producer might just be interested in a ground cover as opposed to a ground cover on a forage. So with that, I'll tie it up, and I'd just like to give a bunch of thank yous to both the GG4GHG team, because they've been funding this major project. So this is led by John Bennett, or co-led by John Bennett, and the teams at the University of Manitoba and Saskatchewan, as well as the AFC team here in Swift Current. My technicians, Katie and Dave, they do all the field work for my program, and they pump through a lot of lines every year. So their work is greatly appreciated. Shanna out in the Salt Lab, we work quite closely on a number of projects. Elira Harrison, who does bioinformatics for my program, as well as Brian James. He was my former technician, but did a lot of work on the prairie clovers. I'd also like to thank my various funders, Genome Canada, Genome Prairie, University of Saskatchewan, Saskatchewan ADF, Alberta Innovates, Manitoba SCAP, ARDAR, Manitoba Ag. So there's a whole slew. And I'm hoping I'm missing anyone there. But with that, I'll just say thank you. If you have any questions about my program or my research, please don't hesitate to reach out to me at my email there. Otherwise, if you're interested in GG4GHG, we recently just put up a new website for the project that's hosted by the University of Saskatchewan, and it's grasslands-genomics at usas.ca. And project updates will be getting posted there as we continue. So with that, I'll finish the presentation and take any questions you have.
Thank you, Dr. Aslan. That was great. Sounds like a lot of very fascinating work going on. Got a few questions in here, but feel free to keep them coming, folks. First one in the chat is there any planned work on seed production for rough fescue?
Yes, so Plains rough fescue, we've been selecting populations here in Swift Current under drought conditions. The Plains rough fescue trial that we're hoping to get into the GBIE testing this year is intended to look at seed production. In terms of the agronomics of seed production, so certain treatments and stuff like that, currently we don't have anything on the docket. Oop, there we go. Sorry, my screen just switched there. We don't have anything planned right now, but that's a good one for like a future ADF project. Dr. Bai at the University of Saskatchewan has done some work in this area, as well as Bill Billigatu, also at the U of S. And I know like someone like Nietzsche would be a good collaborator for that from my perspective. So it's on the radar, but right now we want to test the materials that have been selected for improved seed production to see if that will help before going to the agronomic interventions.
Thank you. Next one, any forage work going on with Canada wild rye?
Canada wild rye, yes. So it's actually, that's a funny question because we've done increases for Canada wild rye for Plant Genetic Resources Canada. We actually do have some new accessions that are going to be going into the ground this year for us to work on. I'm really interested in this species. The challenge with it is a self-pollinated species, so which is good and bad. We don't have a lot in terms of materials so far to start us like to start a breeding population, but we've done a little bit of preliminary work here. We'd likely have to do collections on that species to move it forward, but it is definitely a species of interest. I even worked on it during grad school at the U of M. I've always liked it, so it's on the list.
All right, next one. For the new project combining native legumes with winter cereals, how would you establish and then maintain each crop? Does it require a change to normal cropping practices?
So this would really be intended for people that are already doing work with like fall rye. It's trying to integrate some native species into like a no-till system. So there's different ways that like the living mulch can be established. Our plan is to do a spring establishment, really to give the living mulch kind of a head start, and then going in the fall and then doing biomass removal. So in conventional system, this could be grazing or mowing, but we're going to go in and mow, and then we're going to go and no-till disc in fall rye in August, and then take biomass in the next year. So this is a new project for us. There's different ways it can be done. I mean, there's some systems where both species can be seeded at once. Right now, what we're really interested in is seeing which species will actually work best with the winter cereal, to see where we have that potential to breed and do further downstream work. So, yeah.
Very good. How do your varieties become available to the public? Are there royalties involved?
Oh, yep. Yeah. So how it works for us, we develop a breeder seed, and that breeder seed does get licensed out to a seed grower, and then the seed grower makes it available for sale. So the reason, in terms of royalties, so we don't collect any royalties on materials from like the end user, but if we do have someone that is contracted to do the seed work, they are paying to have access to that seed. So that is where we're receiving our return is from the seed grower. But there's not, if you're referring to like trailing royalties and things like that, that's not something that's on the docket for us. So yeah.
All right. Well, that takes us to the end. Oh, one more came in. What harvest methods are used to get the non-shatter resistant seeds, those missed when harvesting via combine? Are those methods viable at large scale?
That's a really good question, because currently we don't really do too much in terms of treatments to prevent shattering. Basically, it's really timing of our harvest that has been our main focus. On our small plot work, we do bag materials because it's relatively easy to bag a space planted nursery. But for larger scale production, it's really we try to time it the best to minimize shattering. Now, there's trade-offs there. If we harvest too early, sometimes there's issues with the seed not being mature. But if we harvest too late, then we're losing the seed and it's just shattering back into the plot. There has been a little bit of work done in the U.S. The USDA has developed some lines of wheatgrasses where they've integrated a non-shattering characteristic. I don't think that's the method we're going to use down the road for shattering. I think maybe looking at a seed treatment. There are certain sprays that have been used in canola that help prevent shatter. So that might be something that could be employed, but it's not something that we've done a lot of work on just yet. Yeah, we mainly look at genetic variation. That's our big thing. So.
All right. Well, with seed treatments to improve flowability.
Mhm.
When are these added? What
So
do they do to storage ability?
Yeah, so those are actually some of the things we're looking at, like in the winter fat, if viability is impacted by adding these seed coatings. Typically, like the seed coating is added directly, like we try to add it as soon as, or as soon as we can, or how do I phrase this? As close to seeding time as we can. We're trying to minimize the amount of time that we have to have anything touching the seed, because if it's blocking oxygen or doing things like that, it can negatively impact the seed viability. But right now, we're looking at some of those impacts of those seed coatings as part of the project I have with Francois. I know that there's been quite a bit of work done on seed coatings in the U.S. And some work in Eastern Canada. And it really depends on what type of coating is on there. So some groups use like a simple like sugar solution. Others use different types of clay. Others use biodegradable plastics. So and each have their pros and cons. But typically, like I'd recommend, before seed, like adding it before seeding. But you don't want to treat it and let it sit forever in case there's an impact on seed viability. But we don't know yet. It depends on the treatment.
Perfect. Well, I think there's a few more things for Lee to say in the outro, and we're coming up on the hour here. So thanks again, Dr. Aslan, and I'll pass it over to Lee for the ending.
Sounds good. Thanks, Ross. Thanks, Sean. Thanks for sharing your research and all the projects with us. Just kind of some last minute housekeeping, I guess, for today. Sean's presentation will be posted on our website soon. So if you would like to go back or refer back to it, it will be there. As well as you can check out our website for all the other resources for working in native grasslands that we currently have there, as well as you can sign up for the newsletter to be kind of kept up to date with some of our events. Our next event is March 26th, which is in Cochrane, which is the Plant Communities and Recovery Strategies training course. So you can still sign up for that if you're interested. And our next and final webinar for the winter is April 15th with John Acorn. And there was a link posted in the chat at the beginning. At the GRF, we work really hard to kind of keep everything affordable for professionals and stuff. So if you do feel called to make a donation, there is you can donate through Canada Helps. There's a link in there, as well as at the various events that we show up at. You can also purchase swag, which I'm wearing today, the hoodies and shirts and flash drives and all that kind of stuff. I think our next event that we will be at with a booth is the Native Plant Council workshop on the 2nd of May in Calgary. And we will also be at the Prairie and Conservation and Endangered Species Conference in Saskatoon on May 5th to 7th. So you can check out our resources stuff there. We'll have a bunch of stuff there as well. Yeah, with that, thank you everyone for joining today. And thank you again, Sean.
All right. Thanks, everyone.
Yeah.
And if you have any questions, feel free to shoot me an email anytime. So
awesome. Have a good afternoon, everyone.
You.